Have you ever actually stopped to wonder how 4.6 million individual cells just know what to do?
Oh, it's mind-boggling.
Right. I mean, starting from basically nothing, how do they know exactly where to go, who their neighbors are, and what to become to build an entire human body?
It is the ultimate architectural mystery, really.
Exactly. So today we are taking a massive deep dive into the Human Developmental Cell Atlas, or the HDCA for short.
Yeah, and this is such a groundbreaking source we're looking at today.
It really is. It's not just a list of parts. It's a comprehensive, highly detailed map of human prenatal development.
A literal spatial map.
Yes. We're exploring an integrated single-cell and spatial omics atlas. And the sources we're diving into track development from 4 to 22 post-conception weeks.
Which is that critical, super dynamic window where everything is forming.
Right. And they use these massive single-cell RNA sequencing data sets combined with cutting-edge spatial transcriptomics stuff like Visium and Xenium.
Yeah, mapping over 4.6 million cells across 21 different organs.
Okay, let's unpack this, because I feel like we need to explain why this is so much more than just a biological inventory. It's the instruction manual for human life.
That's a great way to put it because, for a long time, developmental biology relied on single-cell RNA sequencing, which is great.
I mean, it tells you exactly what genes are turned on, right?
Exactly. You get the whole transcriptome. But there's a fatal flaw. To do that sequencing, you have to completely dissociate the tissue.
Like putting it in a blender.
Basically, yeah. You dissolve the tissue into the cellular soup. So you know exactly what cells you have, but you lose all the spatial context. You lose the XY coordinates.
It's like having a giant, complicated Lego set that's already been dumped out of the box.
Oh, perfect analogy.
Like, you have 400 red bricks and 50 blue bricks, but you have zero idea what they actually build when they're put together.
Right, because you don't have the instruction manual.
Yeah.
But that's where the spatial transcriptomics, the Visium and Xenium tech completely changes the game.
Because it reads the genetic data while the tissue is still intact.
Exactly. We can finally see the cells in their actual physical neighborhoods. And the HDCA resolves about 450 distinct cell types.
Which is a huge catalog.
It is, but here's the kicker. What they found is that a cell's identity isn't just about its own internal programming. It is heavily dictated by its neighbors.
Wait, really? The neighbors change how the cell acts?
Yeah, completely. So to figure this out, researchers used a tool called Niche Compass.
Got it.
Right. And it uses unsupervised deep learning. So an AI looks at intact 6-week embryo data. And it's not just looking at one cell, it's looking at the continuous chemical dialogue between a cell and its local environment.
It's eavesdropping on the cells.
Basically. And Niche Compass identified exactly 114 distinct tissue niches.
Wait, let me push back on this for a second. Because, embryology is an old science. We've been looking at embryos under microscopes for, over a century?
Oh, easily.
So we know the basic shapes and folds. How on earth did we miss 114 completely specific biological neighborhoods until an AI came along?
Because of what we call morphological similarity. Under a normal microscope, a big cluster of cells might look totally identical.
Like just a generic blob of tissue.
Exactly, just a uniform blob. But Niche Compass looked at the RNA, the actual gene expression, and found these invisible molecular borders.
Wow.
Yeah. For example, the AI found this highly specific temporary niche called the ultimopharyngeal body.
The ultimopharyngeal body. Say that 3 times fast.
Right. Physically, it just looks like a tiny little bump near the throat area. But transcriptomically, it is doing something amazing.
What's it doing?
It's the precise origin point for thyroid C cells. It acts like a temporary waiting room, incubating these cells before they migrate to the thyroid later to handle your calcium metabolism.
Oh, wow. So the AI sees this tiny waiting room that just looked like normal throat tissue to us before.
Exactly. What's fascinating here is that it also mapped the exact dorsal and ventral patterning, so top and bottom orientations, in the exact spot where the embryonic eye and ear are going to form.
Wait, before the eye is even visibly there?
Long before.
Yeah.
The local microenvironment is already organizing and prepping the tissue for these highly specialized sensory organs.
That is wild. Okay, so let's do a quick recap for you, the listener, to lock this in. So far, we've learned that scientists mapped 4.6 million cells using new tech that leaves the tissue intact.
Right.
And an AI algorithm found 114 hidden biological neighborhoods. So I want to pose a question to you listening right now. Think about your own physical neighborhood.
Your street, your city.
Exactly. How does the infrastructure around you, the people around you, shape your daily behavior? Because as we're seeing, your cells operate on that exact same principle. Your neighborhood dictates your destiny.
It really does. And if those 114 niches are the neighborhoods, we have to talk about the physical roads and borders of those neighborhoods.
You mean the scaffolding, the connective tissue.
Yes, the
Yeah.
mesenchyme. Or specifically, the fibroblasts that build it.
Now wait, historically, aren't fibroblasts usually thought of as just the generic glue or mortar of the body? Like, just the boring stuff that holds the important organs together.
Totally. That's how it was always taught. But the HDCA spatial data shows that this early scaffolding is anything but a homogeneous mass.
Are you saying there are highly specialized glue varieties?
Yes. If we connect this to the bigger picture, this completely rewrites our understanding of early embryonic development. These fibroblasts are highly diverse, regionally patterned progenitor cells.
So they know exactly where they are and what they're supposed to build.
Exactly. The Atlas isolated early fibroblasts with highly specific genetic markers like ZIC2.
ZIC2 positive cells.
Right. And these ZIC2 cells aren't just everywhere, they are strictly localized to the scalp, the primary meninx, which is the covering of the developing brain and the spine.
So the central nervous system gets a totally exclusive, specialized crew of biological builders.
Exactly. They aren't just pouring generic concrete. And in other parts of the embryo, you have completely different crews, like cells marked by FRZB or HIC1.
FRZB and HIC1.
Got it. So the builders are tagged with specific gene barcodes depending on the body part.
Yeah. The atlas also mapped the extraembryonic tissue, finding 4 totally distinct fibroblast populations just in the umbilical cord.
Wow, even the cord is specialized.
Yeah. And it even tracks active migration, like the lateral plate mesoderm or LPM.
What does the LPM do?
The spatial data literally catches these LPM cells moving across the embryo to build the mesothelium.
The mesothelium. That's the slippery lining around our internal organs, right? So they don't rub against each other.
You got it. And to me, the coolest part is how they proved this wasn't just an algorithm glitch.
Oh, the skin organoids. I loved this part of the deep dive.
Right. So they grew human skin organoids in a basically lab-grown 3D tissue.
And because it's grown in a petri dish, it doesn't have a mother's blood supply or maternal signals. It's just flying solo. Exactly. But even in the lab, this lab-grown skin naturally developed those exact same ZIC2 positive progenitor cells in the exact same spatial arrangements as a real embryo.
That's incredible. It proves that this regional specialization is just deeply hardwired into human DNA. It doesn't need outside signals to know how to lay the foundation.
Hardwired is the perfect word.
Okay, let's pause and summarize this second chunk. We just discovered that the glue of your body isn't generic. It's actually a diverse crew of highly specialized builders using specific genetic codes like ZIC2 to map out your brain's covering, your spine, and your organ linings.
A completely customized foundation.
Right. So for you listening, think about this. If we now know the exact genetic signatures of these specific biological builders, can you imagine how this could revolutionize how we engineer replacement tissues in a lab?
Oh, the regenerative medicine applications are massive. We could theoretically grow the exact specific tissue you need for a graft.
Exactly. But, okay, let's move on. Because once these specialized builders have the scaffolding up, the embryo instantly needs plumbing.
Right.
Blood and fluid management.
The vascular and lymphatic systems, absolutely.
And here's where it gets really interesting to me. Because if the scaffolding is super specialized, a generic blood vessel isn't going to work. How does the plumbing adapt?
Well, this finding completely inverts our traditional understanding of the timeline.
How so?
We used to think that organs develop their rough shape first, and then the blood vessels sort of infiltrate them later, adapting to whatever the organ needs later in fetal development.
Like building a house first and then snake the plumbing through the walls afterwards.
Right. But the HDCA shatters that. It shows that endothelial cells, the ones that form blood vessels, are specializing incredibly early, in the first trimester.
Wait, so the pipes are customized before the house is built?
Exactly. Take the brain, for example. The capillaries meant for the brain start aggressively expressing a gene called SLC7A5.
SLC7A5. What does that do?
It encodes a really important amino acid transporter. By turning this gene on, the tiny capillary is literally altering its cell walls to restrict what can pass through.
Oh, it's building the blood-brain barrier.
Yes, it is locking its doors and setting up adult-level, high-security barricades before the brain is even fully formed.
That's insane. It's building the bank vault door before the bank even exists.
It's completely counterintuitive, and it's not just the brain. Capillaries heading for the heart express POSTN, capillaries for the liver express OIT3.
They already know their destination and are adapting to it in week 6.
Right. And this contradicts a lot of older mouse models, which suggested this vascular customization happens way later.
So in human development, the plumbing actually helps define the house.
Exactly. The vascular specification provides the molecular foundation for the organ to build upon.
Okay, but it's not just blood vessels. You have to manage the waste and fluid, too, the lymphatic system.
Yes, the lymphatics. They also specialize super early using genes like PSO1, FOXO1, and CALO-CRL.
I remember reading that in the source data, and what shocked me was where they found these early lymphatic cells.
The craniofacial region, the human face and head.
Right, at just 6 weeks.
Yeah.
Which is a huge deal, because if you look at mice...
Mice don't develop cranial lymphatics until after they're born.
Let that sink in. Humans develop lymphatic plumbing in their heads months before birth, but mice do it after birth.
The evolutionary difference is massive, and clinically, this timing is crucial.
Because of primary lymphatic anomalies, right?
Exactly. Those are congenital diseases where fluid improperly builds up in the fetus. For a long time, we didn't fully understand exactly when or why these diseases started in humans because we were relying on the mouse timeline.
But now, because of the HDCA, we know exactly when genes like PSO1 turn on in the human head.
Week 6. We've isolated the exact developmental window where these fluid clearance diseases originate.
Wow. Okay, time for a quick recap on the plumbing. So organ-specific blood vessels and lymphatics are customized in the first trimester, way earlier than we thought.
Long before the organs are finished.
Right. And humans develop cranial lymphatics way earlier than mice. So here is my question for you, the listener. How does knowing that your blood vessels were fully customized for your brain before your brain was even fully built change how you view your body's development?
It really makes you realize how proactive the embryo is.
Definitely. Okay, so we have the scaffolding. We have the customized plumbing. The final system we need to wire up this city is the electrical grid.
The peripheral nervous system, the PNS.
Right. And the PNS has a really hard job. It has to snake through all this localized scaffolding and plumbing to connect everything to the brain.
It's a massive integration challenge. And the PNS actually originates from 2 totally different embryonic populations.
Cranial plate codes and neural crest cells, right? NCCs.
You got it. And figuring out how those 2 sources mix has always been tricky. But the spatial data clarifies some really unique human traits.
Like what?
Well, take the trigeminal ganglion. That's the nerve cluster that handles all the sensation for your face. In model organisms, it's a mix of both placodes and neural crest cells. But the HDCA proves that in humans, the trigeminal ganglion is purely derived from neural crest cells.
Oh, wow. So literally, the textbook diagrams for human embryology are wrong and need to be updated.
Yeah, pretty much. But beyond just the origin, the atlas shows a very strict temporal order to how the wiring is built.
A hierarchy.
Yes. Neurogenesis, which is the creation of the actual nerve cells, always happens before gliogenesis.
Gliogenesis, that's the creation of the glial cells and Schwann cells.
Right, the cells that insulate the nerves.
So wait, what does this all mean for the actual nerve trying to navigate? Because if it's born naked, without its insulation, and it has to navigate this incredibly chaotic growing embryo, how does it not get lost? How does a nerve meant for your arm not accidentally wire itself into your liver?
That is the million-dollar question, and the answer is that it relies entirely on the scaffolding and the plumbing we just talked about.
The fibroblasts and the blood vessels.
Exactly. Those structural niches are established first, and once they are in place, they act like biological traffic controllers. They start secreting specific guiding ligands.
Ligands, let's explain that. It's like a chemical signal.
Right. Basically, they act as chemical breadcrumbs. A really good example from the data is CXCL12.
CXCL12.
Right. It's a chemokine ligand secreted by the surrounding mesenchymal scaffolding. The migrating nerve cells express the receptor for it, so the nerve is literally just sniffing out and following a concentrated chemical trail laid down by the connective tissue.
Oh, So it's like a train. The nerve is the train, it has the engine and the power, but it absolutely cannot move purposefully until the scaffolding workers lay down the physical CXCL12 tracks for it to follow.
That is a brilliant analogy, yes. And then, once the nerve finally reaches its destination, the niche secretes another ligand, like NCAM1, which signals the nerve to stop moving and mature.
That is, wow. Okay, wait. This raises an incredibly important question about diseases, right?
Oh, absolutely. Think about congenital neuropathies.
Right, where a baby is born with a nerve defect.
Yeah. Historically, if a patient has a nerve issue, doctors look for a mutation or a failure inside the nerve cell itself.
Which makes sense. If the wire is broken, you check the wire.
But if the nerve's entire ability to navigate and survive depends on the CXCL12 gradient or survival signals like NGF from the surrounding tissue.
Oh. Then the defect might not be the nerve's fault at all.
Exactly. The Atlas strongly suggests that many congenital neuropathies might actually start as a defect in the surrounding mesenchyme, the connective tissue.
So the wire is perfectly fine, but the drywall contractor forgot to drill the holes in the right place to run the wire.
Exactly. If the scaffolding fails to lay the chemical tracks, a perfectly healthy nerve will just get lost or die off because it never got the instructions.
That completely flips how we would troubleshoot these diseases. Okay, time for our final recap. To sum up this block, nerves develop in a strict order, and they are completely blind without the chemical breadcrumbs left by the scaffolding and the plumbing.
They are totally dependent on their neighbors.
So, to you, the listener, if the physical structure guides the nerves, what do you think happens to the electrical wiring if that early scaffolding is built just a millimeter off target?
It causes a systemic cascade of failures. It really proves the core thesis of the HDCA. In human development, form follows function.
Everything is a coordinated dance.
Right. Tissue structure emerges from these highly coordinated spatial interactions between distinct systems. They do not develop in isolation.
And before we close, we have to mention how accessible this is.
Yeah.
Because this isn't just locked in a lab.
Oh, yeah, the Churita web portal.
Right. The researchers integrated this entire 4.6 million cell atlas with the DDGTP disease database.
Which means anyone, a researcher, a clinical geneticist, can go online, take a rare developmental disorder, and cross-reference it with this spatial atlas.
They can see exactly which embryonic neighborhood is failing in real time.
It completely bridges the gap between raw biology and actionable clinical genetics.
It's amazing. All right, as we wrap up this deep dive into the HDCA, I want to leave you with one final lingering thought pulled right from the sources.
Oh, this is a good one.
We talked earlier about that AI niche compass finding hidden microenvironments. Well, one of its most incredible discoveries was pinpointing the exact spatial location of the forming endolymphatic sac.
Which is a tiny microscopic structure in your inner ear.
Right, it's critical for maintaining fluid balance and your vestibular function. The spatial transcriptomics not only found it, but it identified the exact genes driving its formation genes, like RYZ-GRP1 and PIZO1.
Had a highly specific developmental week, no less.
Exactly. So think about the trajectory of this. If we can now pinpoint the exact week, the exact microscopic neighborhood, and the exact genetic drivers where our inner ear's balance center forms, could this spatial map eventually allow us to trace the precise developmental origin of virtually every congenital sensory disorder?
It's entirely possible.
If we know exactly how the neighborhood is supposed to be built, can we eventually learn how to intervene and correct the blueprint before the physical structure is ever compromised?
It's the ultimate goal of regenerative medicine.
It really is. Something fascinating for you to mull over as you navigate your own physical neighborhoods today.